Second, it starts live streaming.
Okay.
Yeah, we're ready to go.
Well, thanks for the introduction, and I just might use that as a bit of a, I don't know, a segue to the whole talk. So in some ways, my involvement of this came about quite by accident. I just see that Kay Berensmeyer has just joined this group. And it was Kay who invited me originally to work, to do some work in the Shawaliks in Pakistan. And we certainly were not anticipating this ending up being an indicator of the monsoon. So in terms of background, in me getting there, I had previously to that, I'd actually been working with Kay in East Africa on some various things, and I sort of had a notion that stable isotopes could be useful in soils. That was from my undergraduate days at Iowa State University. There was a very strong soils department, and I became interested in stable isotopes. And with my work in East Africa, I was trying to figure out how we could use stable isotopes to reconstruct geological environments. And one of the things that I worked on was a model that where you could determine sort of the contributions of C3 and C4 photosynthesis to soil systems. And, that was kind of interesting. Well, Kay said, well, she's working in Pakistan, and there's this fabulous geological sections of paleosols. And so why don't we just see what's there? And so we did. And so that's what really led me to this topic. And I'm going to try to go through some of this. It's been a long learning career for me, learning about, of course, modern soils and fossil soils, and then really trying to understand some aspects of plant physiology. So I'm not really sure what the audience is. I can see there's a number of you from all over the world. I think an awful lot of you are more in the perhaps geological and monsoon context. So I'm going to try to share with you some of the things that have been shared with me and in my understanding, especially with my colleague Jim Elringer, who's been a good educator on how plants actually work. So Peter Clift contacted me about a year ago and said, well, maybe you could talk about some of the physiological and ecological aspects of plants in the context of C3 and C4 photosynthesis as geologists might need to use them. So that's what I'm going to try to do with this talk. So now if I can figure out how to advance, we did this before.
Yeah, it did work.
All right.
It's working.
So one of the things I'm going to talk about, that'll be the theme of this, is C3 or C4 photosynthesis and the history of atmospheric CO2. And what's important here are some timelines that we'll look at as we go through this process. I think the important thing to remember about C4 plants, and I'll talk a little bit more about them in the future as to what they actually are, but let's just start with if you take the genetic record of C4 plants, it appears that they probably go back on the order of 25 million years. They diversified perhaps in several different pulses. As far as I'm aware, there are less than 5 known fossils older than 1 million years that you can actually say this was a C4 plant. And this is using both anatomy and carbon isotopes. There's one that was found some years ago at 12 million years ago. That's currently being contested by some other people, saying, well, the carbon isotopes weren't actually due to that plant, and the anatomy is fuzzy. And if that's true, then there's maybe one fossil at about 1 million years. But one of the problems that we have is really being able to say something about the abundance of these plants. I exclude from physical fossils the concept of phytoliths. Phytoliths are small silica bodies that are formed within many, many plants. And that's because the classification and identification of the phytoliths continues to be controversial and a problem, but I have hopes for it. There's several groups that are really trying to see if we can make better sense of it, and that may give us hints as to their history of C4 plants. One of the things I mentioned earlier is I developed a method to look at paleosols using carbon isotopes. And what this is a function of is the isotope ratio of carbon 13 to 12 in the atmosphere, the percent of C4 photosynthesis, which, as we will see, has a different carbon isotope ratio than C3 photosynthesis, and soil respiration, how much CO2 is being produced by the soil. And the other real problem is the identification of paleosols. And I will have to say, much to my chagrin, that after we developed this method, I was invited to a number of sites in various parts of the world. And when I would visit them, I would say, well, why do you think this is a paleosol? And people would say, well, we don't know what it is, and therefore it's a paleosol. And I found that to be disappointing. The worst example of this that I found was one where we actually had oolites present, and the person was insisting that oolitic limestones were paleosols just because of the cementing features. So the real problem is in the identification of paleosols, but they've been very, very useful, as we'll see somewhat in this story. And then the other thing where this is recorded is in the diets of animals, and that's because tooth enamel is very resistant to diagenesis. It's already large crystals that aren't particularly interested in recrystallizing, and so they often retain the carbon isotopes as a trace substitution, the CO3 carbonate ion substituting for in one of the hydroxyl positions in bioapatites. One of the reasons that I looked at that was that this would actually attenuate the signal. It might make it, it might actually in some cases make it stronger, because animals would actually choose to eat certain things rather than other things. And anyone who has children knows about that animals choose their diet choices. And so we may be able to enhance the signal. So let's look at where we are. As Jim Zakos 20 some years ago published this nice figure of carbon, benthic carbon through time and oxygen isotopes through time, and gives kind of a schematic of different things. And one of the things that he put on there was this: around 7 million years ago, the expansion of C4 grasses. And there's a bunch of other important things that certainly take place on land. Horses diversify sometime before that. And so there's all of these biological events. A couple of years ago, he published a revision more detailed version of that. Now it's tilted on its side with time going to the right. And the benthic delta O18 record and the benthic delta C13 record as well. And of course, there's the interesting early spikes. The biotic events has disappeared from this figure, although he points out that there's this interesting late Miocene carbon isotope shift that also takes place. So what are the C3 and C4 photosynthetic pathways? There's a basic fundamental difference in the physics of the way that CO2 is fixed by C3 and C4 photosynthesis. So this represents the cross section of two different leaves. A is a C3 plant and B is a C4 plant. And the bright green areas are areas where CO2 is fixed in the plants. And what you see in the C3 plants is that there's a small hole at the very bottom of the diagram called a stomata. The CO2 leaks, diffuses into the plant, and is actually fixed anywhere in this plant, and the Rubisco cycle takes over, and CO2 is absorbed and oxygen is released. That's all we need to worry about from a geochemistry point of view right now. But it takes place in the C3 plant basically everywhere between the two boundaries of the cell, between the two epidermis boundaries. So any of the mesophyll cells, either the palisade cells at the top or the other cells near the bottom, that's where CO2 is fixed in the Rubisco cycle. The C4 plants do something quite different. CO2 diffuses in, and it's fixed as malic acid and is transported to the interior of the cell. So the actual CO2 photosynthesis takes place only inside these, what they call these bundle sheath cells. So in the internal part of the cell, CO2 is actually released in those cells, and the concentration of CO2 in the bundle sheath cell is actually something like 2000 ppm. So that the C4 photosynthesis has a CO2 concentration mechanism that actually concentrates the CO2 in the bundle sheath cell, and that's where the CO2 fixation actually takes place. So this is the biochemical schematic. In C3 plants, everything happens in the mesophyll cell. In C4 plants, the mesophyll, the CO2 is absorbed in the mesophyll cell, made into a C4 acid that's transported to the bundle sheath cell, and then in the bundle sheath cell released as CO2, and the Rubisco cycle then takes place only within the bundle sheath cell. So there's this physical separation between CO2 absorption and CO2 fixation. Well, the net result of that turns out to be very different carbon isotope values, and I'll just touch on that again briefly. But if we look at the tropics, where we have lots of C3 and C4 plants, we can see the sort of the difference between these. And on the left side, we see the delta C13 of C3 plants is at about minus 26 per mil or so, and C4 plants is around minus 12 per mil. And I just show two examples of ecosystems dominated by either C3 plants on the left or C4 plants on the right, where the grasses in the foreground are using the C4 photosynthetic pathway. And basically all trees and shrubs, so woody, almost all woody plants, use the C3 pathway. Many herbs also use the C3 pathway. So just example in your garden, the cucumbers and those kind of things. And in this diagram on the left, the lower story plants, actually, many of those are using the C3 pathway. So C4 plants are not exclusively, but predominantly the tropical grasses and sedges. There are a few other plants using the C4 photosynthetic pathway. So it's present in about 20 different plant groups, but only does it make up a significant component of primary production in any ecosystem as the grasses, or in some cases as sedges. And for those of you who don't know sedges, a sedge is looks like a grass. It has pretty much the same form, but is in more the sort of wet areas. All right, so what we do know is that C3 plants are an early photosynthetic pathway. Kate Freeman and others have identified biomarkers back to more than 2.5 or perhaps 3 billion years that show that C3 photosynthesis was going on. So the rubisco has been around a very long time. Again, to repeat myself, it's tropical grasses are C4 plants, and most dicots, cool season grasses, and are C3 plants. What is significant for C4 plants is that in general, the average temperature of the monthly growing season tends to be higher than about 20 degrees Celsius, and we need to have precipitation during those periods of time. So basically, you need to have a warm growing season. And just to put it into context, when you go to the grocery store, most of the foods and the vegetables and the fruit part of the grocery store are C3 plants. And then, of course, beans and so on would be C3 plants. The cool season grasses, wheat, barley, and rye, so those which, for instance, grow very well in Europe and in the northern reaches of Asia, are C3 grasses. But in terms of important foods, maize, sorghum, sugarcane, millet, teff, fonio are all are C4 grasses. And then animals, if they eat C3 or C4 plants, they inherit that dietary signal. This is just a schematic diagram, just kind of estimating global primary productivity of terrestrial ecosystems. It's estimated that the primary productivity of C4 plants is about 25%, but almost all of that is in the tropics and subtropics. There's very few C4 plants at higher latitudes higher than 35 or 40 degrees. And the other thing that we'll see in this talk is that, and we might think of today's planet as a C4 world, where C4 productivity in the tropics is probably 50% of primary photosynthesis in the tropics and subtropics is due to C4 photosynthesis. This world, the C4 world, we now know has only been in existence for on the order of 6 to 8 million years. So this is just a schematic diagram. We go back, and this helps illustrate what we use. C4 plants have a very narrow delta C13 value. And I will point out, for those of you who aren't the isotope people or ecologists, is within that narrow histogram, there's actually a lot of ecology that the that the difference between being on one side of that that spike in the C4 grasses actually has important ecological implications. And the other important thing to remember is that this whole diagram is shifting to the left as we increase atmospheric CO2. So an important thing to do in considering C3 and C4 plants today in isotope space is to probably to correct those values back to the pre-1750 atmosphere value, because it's been shifted by about 2 per mil due to atmospheric CO2 changing due to anthropogenic activities. But the important thing here is that animals eating C4 plants, as these African Cape buffalo, will inherit a C4 signal, and animals eating a C3 plants will inherit a C3 signal. And we've actually spent a lot of time working with animals, in part because that's easier to interpret than the paleosols, and certainly way less work, because the paleontologists have been collecting fossils for 150 years, and we can use those fossil collections. And I would never, I would want to go to the field and see a paleosol section. I would never accept something in the mail and analyze it without really understanding the field context of individual paleosols. So why is there the difference? One is that the main difference is that C3 plants, if we consider an open or a closed chemical system, C3 plants are in an open chemical system where the rubisco enzyme can choose which isotope of carbon it uses, and it very much preferentially chooses the light carbon isotope, and hence it's about almost 20 per mil more negative than the atmospheric value, which today, of course, the pre-industrial value is about minus 6.5 per mil, and so the C3 plants exhibit that. And again, like with the C4 plants, that histogram, there's a lot of ecology buried in that field of C4 plants, with more drier environments being on the right side of that purple area, and more mesic areas on the left of the purple area. Anyway, the C3 plants are allowed to choose which isotope they want to incorporate, and as a result, they're about 20 per mil depleted in carbon-13 compared to the atmosphere. C4 plants actually do their photosynthesis in a closed system, and isotope discrimination is not allowed. So there's a diffusion effect of about 4.5 or 5 per mil that shifts from minus 6.5 or minus 7 per mil to about minus 4.5 or minus 5 per mil to the left. And C4 plants basically in this closed environment, they have to photosynthesize. They don't get to choose which isotope gets used, and so there's technically there's no carbon isotope discrimination. But when you get into the details of talking to a plant physiologist, you can find out, oh, there are ways in which you can have a minor amount of carbon isotope discrimination, and that broadens the peak of the C4 delta C13 values. So it's really an important difference: open open system versus closed system discrimination. So not going to the paleosols, but looking, but the paleosols was where we first noticed this difference. But we decided then to try to work on the diets of animals because we would get this slight enhancement, and it's much easier to than having to do the field work in so many different paleosalt areas. So one of the things that when we got into this that we expected, and this is a lesson to the younger scientists, we very much expected that we would find, so we found a huge isotope shift in Pakistan at about 7 million years ago. And we thought, okay, well, now let's go to North America, and it's going to be spread out over about 20 million years, because as pointed out in that first diagram by Jim Zakos, equids, horses, became hypsodont. Their teeth became hypsodont starting about 20 or so million years ago. And the paleontological story going all the way back to G.G. Simpson is, oh, well, even older than G.G. Simpson. Oh, they got hypsodont because they started eating grasses. And what kind of grasses? Well, the same kind that they're eating today. The kind they're eating today is C4 grasses. So we expected to find in North America, where there's a wonderful fossil record, that we would find C4 evidence of C4 diet back at 10 million years, at 15 million years, and 20 million years. And lo and behold, nothing. We got nothing until about 7 million years ago, which is strikingly close to the time that we'd identified this in Pakistan. Wow, this is like possibly a global event. So then we also, I was working in East Africa with Miv Leakey, and we said, well, let's just quick look in Africa. Oh, lo and behold, same time, about 8 million years ago. And we then worked with some paleontologists in South America. Oops, same thing in South America. So we seem to see this something approaching a global event. If we look at the details of it, it looks like it could have been a little bit earlier, shade earlier in the tropics than the subtropics, and so on. Anyway, it's very robust in very different places in the world by 7 million years. So this was some of the early Pakistan story. In Pakistan, in paleosols, we found this very surprising change, and then we looked at the diets of animals. So the top diagram is Pakistan writ large, where we've analyzed many more samples than our initial samples. But again, we see that there is an enhancement in the diets of animals that makes it easier to find the earliest signals. So we have this expansion of C4 biomass globally in the late Miocene. So what's the reason for it? I just want to go into this because I think this is some important lessons that I've learned in talking to my biological colleagues. And let's just consider that there's two reactions that Rubisco can do. The photosynthesis reaction, which has been around for 3 billion years, CO2 is fixed, oxygen is released, okay? What's less well appreciated is a reaction called photorespiration. During photorespiration, the wrong reaction occurs. It turns out Rubisco can take up oxygen. And when it does that, it releases CO2. Okay, so you might think of this as a good reaction and a bad reaction. And it turns out both reactions are functions of temperature. And so really this is a competition between the oxygen reaction and the CO2 reaction. And Rubisco much prefers the CO2 reaction, but maybe what's really important is the ratio of oxygen to CO2. So the top diagram, let's go to the bottom diagram. The bottom diagram is C4 photosynthesis, and there is no bad reaction taking place. Photorespiration but just doesn't happen. In C3 photosynthesis, we see a similar reaction rate. There's a increasing reaction rate, but the C4, the bad reaction, the photorespiration reaction, actually increases at a higher rate with respect to temperature than the good reaction. And so eventually there's a crossover, and the final crossover is way over on the right, where actually you have no net photosynthesis at all. So what I've tried to illustrate in this diagram, in these two diagrams, is there's a point where C4 photosynthesis becomes favored over C3 photosynthesis, simply because too much of the bad reaction is happening in the C3 plants, and you never get the bad reaction in the C4 plants. So what I'd like to look at now is some monitoring that I did with one of my students, Will Mace, some years ago. And we monitored the temperature in soils at 3 sites. So this is an open grassland, a bushland, and a floodplain on the lower Tana River in Kenya. And we monitored these sites for a year, the soils, and from that we calculated the instantaneous hour by hour ground surface temperature of the site. And December, January, February is a dry season in this area. So we basically, this is the skin surface temperature. And we see that in this open grassland, the skin surface temperature on average reaches 60 degrees Celsius, which is pretty darn hot. In the bushland, in that middle figure, we get temperatures in excess of 40 degrees Celsius. And then in the riparian forest, the middle of the day, it might get up to into the 30, on the 32, 34 degrees Celsius. Well, what does this mean in terms of photosynthesis? 40 degrees Celsius is just too darn hot. And so in the open grassland, we only have photosynthesis during part of the day, and even in the bushland, we only have it during part of the day. So it's important to consider these temperature relationships, because if it's too hot, we don't have the photosynthesis. So let's look again at the CO2 and the oxygen, and think of this in terms of the competition between oxygen and CO2, and what is the ratio of oxygen to CO2 during photosynthesis. Well, oxygen has a concentration of about 200,000 ppm in the atmosphere. And for a C4 plant, where you've got this concentration mechanism, oxygen reaches a concentration, or CO2 reaches a concentration of about 2,000 ppm because of the concentration mechanism. So the oxygen to CO2 ratio is about 100 in a C4 plant. In a C3 plant, that doesn't happen. The oxygen is still 200,000 ppm, but CO2 is actually lower than atmospheric CO2. So say CO2 concentration of about, I forget what I've calculated here, but even at a modern 300 ppm, CO2 or 400 ppm, CO2 inside the plant is 100 ppm lower, so it's about 300 ppm. And so that ratio now is around 700, not 100. So oxygen, so it's much more difficult for the rubisco to find that elusive CO2 that it needs for photosynthesis. So that's the story. We've got this concentration mechanism in the bundle sheath cells. CO2 at 350 ppm gets elevated to 2,000 ppm, but at 350 ppm, sort of when I was in graduate school, the internal PCO2 was around 250 ppm. And this gradient in C3 plants seems to hold fairly well, about 100 ppm gradient between atmospheric CO2 and what's known as CI, to the ecologist, internal PCO2 in the C3 plant. Well, this is what happens to the ratio as we change CO2 concentration at wherever site we're talking. This is the oxygen to CO2 ratio. It goes from really high values to really low values. And so perhaps one of the important things to consider is what is the history of atmospheric CO2 over geological time. We know that very early times in 3 billion years ago, when Rubisco evolved and invented itself, CO2 was the dominant gas in the atmosphere and oxygen was the trace concentration. The roles were reversed. CO2 was dominant, oxygen was a trace gas. Today, oxygen is dominant and CO2 is a trace gas. So oceanography has provided us with lots of information of climate change. This is just a nice diagram from Lezecki and Maureen Ramo showing the changes in the obliquity cycles as recorded in marine sediments. Then there's a number of ways in which people have tried to use to reconstruct atmospheric CO2, and I'm just gonna summarize some of that. I know that Barbara Hernisch is doing a new compendium, and I'm not going to quote that because that's unpublished, but we'll just take some of the published data. Here's some work from Tripathi trying to reconstruct atmospheric CO2 using boron isotopes. And what we see is that it seems to work during the Pleistocene, the latest Pleistocene, when we know from the ice cores what atmospheric CO2 oscillated between 180 and 280 ppm. And the early Pleistocene seems to be a bit higher. And so here's Ray's CO2 reconstructions based on boron isotopes. Through most of the tertiary, well above 500 or 600 ppm, and falling below 400 ppm sometime in the latest Neogene. Looks like it crosses the line here around 9 or so million years ago. And then we're in this period of low atmospheric CO2. So again, taking Tripathi's more detailed data, we can look at atmospheric CO2 through the last 20 million years and consider that, well, if oxygen is essentially constant at 200,000 ppm with CO2 a variable, we can see that over the last 8 million years, we've gone from down here in the right in the yellow area where we're at oxygen to CO2 ratio in the atmosphere is around 500. And you can see it's been increasing. And this would be the stomatal oxygen to CO2 ratio, where the internal CO2 inside of a C3 plant is 100 ppm, always 100 ppm lower. We see that if there's a threshold in there, perhaps we've crossed it. So that's what I'd like to leave you with, is that the idea that perhaps there's a important threshold that's crossed that favors the C4 photosynthesis. There's going to be a bunch of other factors that are going to be playing into this. And I was just talking in the beginning with our host about this. Aridity is going to, and seasonality is going to be important. C4 plants that have a bunch of strategies that actually makes them favorable to fire conditions and drought conditions by having their reproductive organs often underground. So that helps them in those situations. So I'll leave you with this thought. Is there critical thresholds? And it could be that this expansion of C4 biomass is related to this critical threshold. And I'd like to just leave you hoping there's some oceanographers and modelers out in the background, is there's this intriguing shift of late carbon isotopes of about 0.5 or 0.6 per mil at about the same time that we have this global expansion of C4 biomass. Some years ago, I did a back of the envelope calculation and calculated that changing from C3 to C4 biomass in the late Miocene actually can result in a carbon isotope shift about half of that effect, about 0.3 per mil in just due to changing from 100% C3 into this C4 world. So I think I've tried to give enough time in this talk to leave time for questions, and so I'll close with those two ideas. Thank you.
Thank you so much. It was a tour de force reminding us where we stand, and you're ending with the delta question. I've always wondered myself about that. It's not much discussed in these compilations, and it seems to me like a chicken and egg question. So I'd like to open the floor for questions. Please do it either way. Ask the question, raise your hand, send it to me, any way you want. If there are no questions, I'd like to ask one. Oh, Barbara Carapa, please unmute yourself and ask the question.
Hi, Terry. I really enjoyed your talk. Thanks. So the question for me is maybe the big one is, what do you think is then driving this global shift in C3 to C4 plant? Because, for example, as you well know, and this comes mostly from your work in the Central Andes, that corresponds to increased aridification. But in other places like Pakistan and the Himalayas, that's intensification of the monsoon. So can we find a commonality in terms of shifts in paleoclimate that will result in shifting ecology?
I think we need a couple of things, and that's to realize that there probably are cofactors, but the cofactors might be buried inside understanding really some of the anatomical issues and some of the physiological issues of C3 versus C4 photosynthesis. Unfortunately, we just don't have any fossils of C4 plants. And this has to, in my opinion, represent one of the most important terrestrial changes in plants. It's equivalent to the rise of the angiosperms or the rise of the land plants at all. So we've got, some maybe 20 million years of evolution, which we know nothing about what actually, happens. What we do know is that certain, not all C4 plants, have this strategy of being able to propagate readily as an adaptation to fire by putting their reproductive mechanisms, concentrating those underground. But we don't know when that arose, and we don't know really how important that is. we do know that fire globally changes at around 8 million years ago, and certainly in the Pakistan region. So that could be a cofactor. What favors fire? Well, certainly increased seasonality does. I'm always hesitant about this word aridity because it means such different things to different people. I grew up near Chicago. Chicago gets a meter of rainfall a year. It's dominated, the natural vegetation is almost all C4 plants. Is that arid? I wouldn't call it arid. Likewise, Nairobi gets a meter of rainfall a year. I wouldn't call it arid, so I'm not sure what aridity means. And so I haven't answered your question, but I think the seasonality, how that affects fire, and this other driver of oxygen to CO2 ratios, all of these are important and will have different effects in different places.
So, Thure, do you think this the physiology of plant is the problem, or our records, the proxies for records? I mean, the physiology seems pretty clear. We just don't know how to describe the degrees of freedom in all these directions: fire, seasonality, aridity, that I would think is the average of seasonality. Do you think that our proxy records are what we miss, besides the fossil, which I agree?
Oh, definitely. We're working on proxy records of aridity, and I think that 017 is going to be a big help there.
Okay.
Yeah, and I think we need better understanding again as to what when we use the words, like especially when you use the word aridity, what is it that we actually mean? And
yeah,
the seasonality is clearly going to be a major thing. And how in the world do we develop proxies for both of those, the aridity and the seasonality, and we need further ones for fire.
Yeah, I see another raised hand from Kay Berensmeyer, please.
Yeah. Yes, thank you. Great talk, Thure. I enjoyed it a lot. It occurs to me that you mentioned the genomic record puts the origin of C4 plants back to about 25 million. And I guess we need a grass expert on this talk, but you would expect that there would be quite a diversification of C4 grasses based on this new physiology. And even if we don't have a fossil record, it seems like tracing the genetics system of the crown group of grasses back and looking at what and where was happening with the diversification would be another interesting line of research, and probably has been done, including resistance to fire.
Yeah, I don't know if that's been done. What I just tend to think there are not quite enough conversations between people developing the proxy records and some of the ecologists and genetic people. I know that I have a hard time following some of the genetic arguments and kind of have to take their word for it. But I think we just kind of need a little more education and perhaps a little less dogmatism about things. But yes, I think the genetic records are going to tell us things. We need to be able to ask the right questions and be able to also have an appropriate degree of skepticism, and that's often hard to communicate properly.
Thank you. I see another hand for Catherine Badgley.
Yep.
Please.
Right. Thanks, Thure. I always learn something new from you every time I hear a talk. So it's great to have sat through this one. My question is a little bit forward-looking. Given the fact that you've revealed these essentially new questions that we need to answer based on the fossil record, potentially the phylogenetic record, what kinds of new data should we be looking to gather from either mammalian teeth, from paleosols, from biomarkers, or from any other source that you think might help us continue to understand the evolution and the increase in C4 ecosystems across the terrestrial world?
I think continuing to make sure that you have a solid foot in understanding the proxies that you develop, and really trying to test them yourselves rather than waiting for 10 years for somebody else to show that you were wrong. I think, this particular work that I've outlined here really suffered, I think, because the early alkanone work said that no, CO2 was much, much higher, and some of the people who developed the alkanone work, Brian Pope in particular, was saying, no, this alkanone work is not as robust as you people are saying it is. And it turns out he was right. It was overestimating PCO2, and that's just the way that it. But I think we need more skepticism of the proxies as we develop them, because we're so anxious to use them in the geological record. But we need more work there. I'm kind of hopeful that that the phytolith work is going to bear important fruit. But again, with that, I really want people to be appropriately skeptical as they develop the proxies.
Right. Thanks.
Thank you very much. I really think that put the dot on the I on the proxies thing. I'm thinking about the alkanone records saying that there is no Miocene CO2, high CO2, as an example. I don't see any other questions. Please raise your hand or intervene. If not, if there are no questions, I was honored to listen to this lecture, and your sharing thoughts on the future of this field. And I thank you very much for joining this seminar. Thank you, Thure.
Well, good. This has been a lot of fun, and yeah, put me on the mailing list. I'll try to come to some of these other future talks as well. So
you could also select from 2 years worth of talks if you would like to see what it is. It's available. So thanks again. It was really great. Thank you for joining us.
okay. Bye-bye.
Bye.